we have, and then it will be a summer break, and we will start in September. Ms. Palzeska, are you paying attention when we're starting?
Yes, we start on September 5, and the next week...
September 5, so that's when we were going to start again. So
we still have the next week seminar.
So again, thank you very much. It was a wonderful year of great seminars, and today is not going to be exception. So welcome everybody here and as well as on Zoom. And let's celebrate vision research. And with that, I will ask Roman to introduce our speaker.
So good morning, everyone. It is my great pleasure today to introduce Dr. Douglas Dean from University of Louisville School of Medicine. Dr. Dean acquired his PhD degree in 1983 from Kansas University Medical School, then continued his postdoctoral studies in Baylor College of Medicine, and later from 1986 until 1988 at Salk Institute. Between years 1988 to 2005, he held several positions at the Washington University School of Medicine in St. Louis as assistant professor and professor of medicine and cell biology and a member of molecular oncology program. Currently, he serves as Robert and Gretchen Ranzavel Professor of Ocular Oncology at the Department of Medicine and Ophthalmology and as a member of Brown Cancer Center at the University of Louisville School of Medicine. For his research, he received numerous awards, including Newmark Award for Biological Research from University of Kansas, Spirit of Health Award from American Cancer Society, or Wasserman Merit Award and Stein Innovation Award from organization Research to Prevent Blindness. Dr. Dean contributed to the scientific field with significant achievements. He and his group characterized the regulation of transcriptional inhibition mediated by tumor suppressor retinoblastoma protein and provided the first evidence of its direct interaction with the epigenetic modifiers such as HDAC or HPC2 to establish its dual mode of action. His later studies described metabolic processes in the RPE and the retina during retinitis pigmentosa, leading to degeneration or potential restoration of photoreceptors. More recently, he investigates the role of mesenchymal transition linked to transcription factor ZEB1 as a potential therapeutic target for pathological vascularization and inflammation in the retina. His research was published in high-profile journals such as Nature, Cell, and Science with a total number of 171 publications and over 17,000 citations, and is supported by several NIH grants. And now, without further delay, please join me in welcoming Dr. Dean and with his presentations on metabolic changes during retinitis pigmentosa progression.
Well, thank you for that kind introduction. I hope that, with all of the quantity and quality of research coming out of UC Irvine, that I can at least make a small contribution today. I'm going to talk to you about retinitis pigmentosa. As you all know, this is a disease that mutations in multiple different genes wind up giving at least a somewhat similar phenotype where there's loss of rod function, then loss of rods, and ultimately diminished cone function. It's really that cone functional loss that I want to talk to you about today. Let's see. Yeah. So in this slide, what I really want to do is sort of sum up a couple of approaches we're going to take. We're going to look at today mostly a pig P23H rhodopsin model. I'm going to sprinkle in a little bit of mouse P23H rhodopsin model that you guys are very familiar with. And I'm going to talk to you really about two major approaches that we're taking to try to get at metabolic changes that are occurring here. The first is illustrated up here on the top, and it's really dependent upon Robert James, who's a vascular surgeon, and his fellows who run catheters in very small places and manage to get it up into the ciliary artery. And we look at metabolite introduction. There we collect tissues and look with LC and GC-MS at changes. Now, another approach is really kind of stolen from Rabinowitz's group. They published a few years ago in Cell Metabolism that there are multiple changes, metabolic changes in different tissues in the pig. And basically what they did was to collect blood from arterial flow into the organ, and then from veins draining the organ. And they looked at differences to try to highlight metabolism in that organ. And basically what they found is that, collecting blood, arterial blood from multiple different sites in the pig gave a fairly similar metabolic profile, but it was as you might expect, it was the veins draining the different organs that really reflected uptake or secretion of metabolites from the organ. And they were able to look at a variety of different organs. And we tried to use the same procedure to look at the eye. And basically what we've done is look at blood collected from the ciliary artery, the ear artery, even the femoral artery. And just like Rabinowitz's group, we found fairly similar metabolic profiles in the arteries. And then we looked at the vortex veins, which in the pig, there's 4 of them that drain the different quadrants. And basically by comparing this, we tried to get some idea of what's going on metabolically in the eye in wild type versus mutant animals. Doug Emery and Eric Vigmanic are responsible for all the blood collection as well as the ERG studies that I'm going to show you. Wei is a retinal surgeon, and she's responsible for injection of outer segments and some outer segment mimetics that I'll show you. Xiaoqing and Yanqing are really responsible for logistics of the pigs, doing some of the immunostaining. Julia is invaluable in looking at sort of raw single-cell RNA sequencing and converting it into our languages where we can look at it. And then, of course, Jianhai Du is a longtime collaborator that's really responsible for all of the mass spec data that I'm going to show you. So I think you're all familiar with the concept that the RPE is the sort of the blood outer retinal barrier, and in doing so, I've illustrated a couple of things that it sort of mediates transport of things like glucose from the choroid circulation to photoreceptors in the outer retina. I've also put proline on here. The glucose transport is thought to, glucose is thought to be relatively unaltered in its transport to the photoreceptors, and whereas proline is metabolized into a number of different metabolites that are then transported to the photoreceptors. And things like lactate produced by the photoreceptors are then transported back to the RPE, when they may be metabolized into the TCA cycle or even secreted back into the circulation, as well as lipids from the outer segments that are phagocytosed contribute to the metabolism in the RPE. So there's really an integrative environment here between the RPE and the photoreceptors, and many groups have pointed that out. We're going to talk mostly today about glucose, but I don't want you to get the idea that this is the only metabolite that's involved. And what I'm going to try to show you is some of our data looking at maybe changes in glucose metabolism as RP progresses. These are some earlier experiments that we did in the mouse, the P23H knock-in mouse that you guys are very familiar with, because they're really too expensive to do in the pig. And basically what this projects out is just showing the loss of rod outer segments that occurs during progression in the mouse. And you can see it around P35, you're beginning to lose some of the rod outer segments, even though most of the rods themselves are still present. And what we've done here is just inject fluorescently labeled deoxyglucose into the tail vein, and then look an hour later at where it's going. And you can see that at P25, most of you can see glucose in the choroid here, and it is transported into the retina, as you can see here, and it's concentrated in photoreceptor inner segments here. Then by P35, as you're beginning to lose the outer segments, this glucose begins to accumulate in the RPE, and it's diminished in the photoreceptors. Consistent with the idea that these outer segments are important for the glucose transport, if you transplant outer segments from wild type mice into the subretinal space, and these are labeled with a fluorescent dye, you can see that you again promote the transport of this deoxyglucose back to the inner segments. So this led us to suggest that in retinitis pigmentosa that there's a transport problem of glucose from the RPE to the outer retina. And we looked at this in some more detail in the pig as well as the mouse, and the notion is that RPE outer segments interact with integrin and MERTK receptors on the apical surface of the RPE. I think this is fairly well established. Both MERTK especially, but also integrins, signal through AKT. This is important for cytoskeletal changes that mediate the phagocytosis of these outer segments. But this AKT signaling also classically regulates the transport of GLUT1 and GLUT4 from the cytoplasm to the surface of cells. And mechanistically, these transporters are held in the cytoplasm by TXNIP, which is perhaps the most glucose-inducible gene. And in response to glucose, it negatively feeds back to hold these receptors in the cytoplasm. This is really the major target of metformin, which allows transport of GLUT transporters to the cell surface and actually promotes glucose uptake in type 2 diabetes. So as I showed you, there's a correlation between rod outer segment loss in RP and a failure to transport the glucose from the RPE. In these impolarized cells, frequently there's a partitioning of AKT signaling and also a partitioning of different proteins on the apical and basolateral surface. So what we found here is that GLUT1 is mostly cytoplasmic, but on the basolateral surface, it's still present. And then when we transplanted the outer segments back into the RP pigs, you could see an onset of AKT signaling again, transport of GLUT1 to the cell surface, and movement of glucose into the subretinal space. Now, Finneman's group and other groups have really done pioneering work showing that it's really phosphatidylserine that becomes externalized on the tip of outer segments that interact with integrins and MERTK on the RPE apical cell surface, and this is responsible for signaling. Now, we tried to mimic this with liposomes that had phosphatidylcholine and serine, and when we injected these into the subretinal space, we again, in RP, we again triggered AKT signaling, transport of GLUT1 to the apical surface, and glucose transport, whereas control liposomes with only PC didn't do this. So we concluded from all of this that there is an issue with the transport of glucose to the subretinal space from the RPE in RP, and this is linked to loss of abundant rod outer segments. Now, moving to the pig, we sort of looked at the same kind of thing with mass spec, looking at the RP. At the RPE, you can see that in the RP model that glucose is accumulating in the RP and it's diminishing in the retina. And so then we looked at the metabolites and glucose in particular in the artery and in the four vortex veins that are present. We looked at the vortex veins in 2 different eyes and 6 pigs, and we compared that with mass spec to what's going on in the artery entering here. And we took it that a decrease in metabolites in the vein, indicated uptake into the tissue, and an increase in the vein really export from the tissue. So when you look at this, you can see that glucose is taken up in the wild type as expected. In the RP pig, this is diminished. Then when we inject the outer segments in here, this uptake is restored. This led us to conclude a couple of things. First is that the buildup and the failure of transport of the glucose in the RPE is responsible for the diminished uptake. And then with the outer segment transport and the stimulation of glucose transport to the subretinal space, this import was restored. And the other thing that we concluded from this is that this difference between the artery and the vortex vein was really highlighting glucose import likely into the RPE. Okay, so but in addition to this, if you look at what's going on metabolically in the RPE, there's we think is a sort of a potentially a second issue. One is that if you look at expression from single cell RNA, comparing the RPE rods, cones, here you can basically see that the RPE, is certainly not enriched for glycolysis, which we think allows, movement of most of the glucose through the RPE without it being metabolized. And, consistent with, phagocytosis of the outer segments in the wild type animals, you see enrichment in the pathway for fatty acid oxidation that's occurring. So we think based on this, there's a decrease in enrichment for glycolysis, and that the fatty acid oxidation pathway is enriched in the RPE. Now, just as a little bit of an aside, this is just some data that we published trying to look at different stages in the pig of RPE. So what we've called earlier RP, if you look at this EM, you can see the rods are essentially lost. You still have cone inner segments and outer segments at this stage. And this is around P120, P150 in the pigs. By 12 months or a year of age, the cones persist, but they've lost their outer segments. They still have inner segments. And these cones survive for at least 5 years in the pig. And we've sort of termed this the later stage of RP, at least for these purposes. And we've looked at metabolism of C13 glucose delivered through the ciliary artery here. And of course, the caveat here is we are comparing, retinas with and without rods, which is obviously a major difference. But when you look at that, basically you can see there's still metabolism of the glucose, in the early RP. But in late RP, this metabolism is essentially lost from glycolysis, the TCA cycle, etc. And one interesting thing is that with lactate is transported into the RPE, at least in the pig and under these experimental conditions, we did not see the lactate metabolized into pyruvate or into the TCA cycle. And we can follow it being transported from the RPE into the choroid circulation. So we think, at least under these conditions and in the pig, most of the lactate is being secreted. But interestingly, when you look at the retina and you look at C13 lactate, it's definitely getting into the retina, and it's being metabolized in really sort of a similar fashion in the wild type as well as late RP, which, really the only thing remaining photoreceptor wise are these cones lacking outer segments. So we take this to mean that likely these cones that appear sort of dormant in the late stage RP may be surviving by metabolizing lactate. And so anyway, that's an interesting, somewhat of an aside. But the thing I really wanted to point out is this figure here in panel L and what's happening in the RP model in the RPE with RP. And by the early stages of RP, while you see few metabolites, representative of glycolysis in the wild type, in RP, there's an onset of accumulation of these markers of glycolysis. So we think this indicates an onset of glycolysis in the RPE, and that some of that glycolysis is diverted into the pentose phosphate pathway and in production of NADP, which I'll talk a little bit about in a second, also glycogen metabolism. So in addition to, this is just sort of a model showing, what we think might be happening here in the RPE. So in the wild type with the, with the abundant fatty acids being taken up by outer segment phagocytosis, the gene expression is at least consistent with enrichment of the fatty acid oxidation. And the interesting part of that is that multiple steps in fatty acid oxidation block glycolysis. And then with the decrease in phagocytosis of these fatty acid-rich outer segments, we believe that glycolysis is initiated, and we've even shown evidence for onset of fatty acid synthesis here. So we think that one reason for this onset of glycolysis is just simply the loss of the fatty acids for fatty acid oxidation. If you look at what goes on with fatty acid oxidation, you create a large amount of acetyl-CoA. This is really reminiscent kind of what's going on in the myocardium of the heart. There's several consequences of doing that. One is that you diminish free CoA, which is really important for a number of metabolic pathways. And also the acetyl-CoA can acetylate various proteins and change metabolic pathways. Now, to get rid of some of this CoA and of acetyl-CoA and free up CoA, the acetyl groups transfer to carnitine, and then that can be secreted from cells. And indeed, in the wild type RPE, we see acetylcarnitine accumulate, and we can show it being exported from the RPE into the circulation. Okay, so the bottom line here is we think there are two problems, at least two RPE-centric problems with glucose transport. One is actually the transport of glucose itself, and the other problem is the onset of metabolism of glucose in the RPE and in RP. And both of these combine to really limit the amount of glucose that's available to photoreceptors in the outer in the outer retina. So this is just to kind of hammer home what we're thinking is that we think in the wild type RPE, the metabolism is highlighted, this is an oversimplification, by fatty acid metabolism and fatty acid oxidation. And with the loss of the fatty acids feeding into this, there's an onset of glycolysis. With some of that being diverted into the pentose phosphate pathway, and as well as into the TCA cycle. Now, looking at this pentose phosphate pathway, if you look, this is mass spec analysis of the RPE, in RPE and in wild type animals, and basically what you can see is that what I showed you, there's an accumulation of components of the pentose phosphate pathway, and there was a dramatic increase in NADPH production, which is critical for antioxidant protection. And you can see with the oxidation of the glutathione here in the RPE that we speculate that this NADPH production that's occurring here is really, a protection against reactive oxygen that's potentially accumulating in the RP. Okay, so I want to spend the rest of the time that I have talking about a new function for glucose that we sort of stumbled across that isn't published yet. And to do this, I'll just remind you, we've sort of broken down the RP progression in the pigs shown here to sort of an early stage where you're losing outer segments of rods shown here in blue. You're beginning to lose the rods. The cones still have outer segments. And then in the intermediate RP, most of the rods are lost. You still have cones with outer segments. And then in late RP, your cones have lost outer segments, but these cones, at least in the pig model, seem to persist for a long period of time. And it's notable that this is probably somewhat representative of when most patients are presenting in the clinic, where they show a loss of nighttime vision, peripheral vision, and even photosensitivity, and a sort of an extended time period of regaining acuity, following luminance changes. So what I want to show you here is just some full field ERG, and this is a photopic ERG, and we're comparing wild type to really intermediate RP that's around P120, P150 in these pigs. And you can see with a standard sort of ISF flash that's really 1 millisecond or less that the B wave here, from the bipolar cells, very similar still in the wild type and the RP pigs. And this B wave is rapidly brought back down to background, and this is maintained in a longer duration flash, even out to 1 second. And bringing this back down is critical for for the pathway to be able to sense luminance changes, which is the hallmark of cones, and also for visual acuity. So the interesting thing that we found is in the RP, although it looks similar initially, with the longer duration flash, there's a failure to bring this signal back down to baseline, and this just accumulates. And you see with a light offset, you see a D wave, indicative of an off pathway signal. Now, looking at this, we thought, well, this could be either, a failure to suppress an on signal, or potentially loss of an off of the cone off pathway here that's causing this. And so when we treated with APB to block the on pathway, and basically found that the wild type and the RP were very similar. There's still the D wave. So we concluded that this is likely due to a failure to suppress the on pathway with a long duration signal. Try to go through this quickly, but we also looked at going back here at luminance changes. We reasoned that although under these conditions the signal is being brought back down quickly to zero, but as you increase the luminance of the flash, we found that just like with a longer duration flash, the RP pigs failed to bring the signal back down even to a brief flash, where the wild types did. And then we looked at flicker, and as you know, this is really just a measure of the ability to reestablish a baseline in preparation for a new rapid flash. And this is a series of 30 flashes or 30 trials that we're looking at. And when you look at the wild type, you know, it's pretty similar in the early trials all the way out to the late trials. But when you look at the RP, although the early trials appear to be at least somewhat similar, obviously they're not exactly, to the wild type, as the trial number, you know, progresses to the late trials, there's really a problem in you know, in duration of the signal and the signal height. So based on all of this, we concluded that that in the RP, that the on cone pathway signal, really there's a problem with exhaustion of the ability to bring this back down to baseline when you either have an increase in luminance, an increase in a duration of a flash, or when you challenge the animals with multiple repeated flashes. Okay, so you know what might be causing this? So classically, you know, signals are positive signals are regulated by inhibitory neurotransmitters such as GABA and glycine. And when we looked at with mass spec at the retina in these animals, we saw that GABA levels were greatly diminished compared to wild type, whereas glycine really wasn't changed. And if you looked at immunostaining for GABA here in the retina, you can see that with immunostaining, just like with the mass spec, there's diminished GABA in the retina. And when we ask, well, you know, what are the targets of this? What targets might GABA be hitting in the retina? And we looked here at RNAs for different GABA receptors, and you can see that the on bipolar cells as well as rod bipolar cells express GABA receptors, whereas the off bipolar cells really have much less expression, suggesting why the on pathway might be targeted by GABA, and with reduced GABA, this pathway may become deregulated. Okay, so with this, not unexpectedly, when you look at what the source of GABA might be in the retina, looking at single cell RNA expression and dot plots, basically you see not unexpectedly that that it is the GABAergic amacrine cells that are really expressing the GAD1 and GAD2 genes that catalyze GABA production from glutamate, and that really none of the other cells in the human or in the pig really express much in the way of GAD1 and GAD2. This is not exactly the case in the mouse, but in the pig and the human, that seems to be the case. All right, so this led us to this kind of a model, or this kind of an idea, that the GABAergic amacrine cells are targeting GABA receptors on the bipolar cells. This is known to trigger an increase in chloride transport and polarization that really blocks the signal. But with the reduced GABA that in RP, really this is lost, and you really have a constitutive on signal that's occurring in the cells. Now, when you look back at the brain and what's going on there, there's a classic loss of GABA signaling in patients that lead to deregulated signaling in the brain. This triggers seizures, this triggers sleep disorders, and it triggers anxiety. And this can classically be treated in a couple of ways. One is by drugs like vigabatrin that target GABA turnover and would lead to increasing GABA levels in the brain. The other is benzodiazepines, specifically lorazepam or Ativan. These bind to GABA A receptors, sort of open them up so that when GABA binds, there's an increased transport of chloride through these channels, and in that way, they sort of super activate these GABA A channels and compensate for a low level of GABA. So we wondered about this in the pigs, and we used bicuculline, which specifically targets GABA A receptors. And what we found when we compared the wild type, the untreated and treated, that there was a big increase in the bipolar signal and also somewhat of a delay. But this didn't look exactly like what we were seeing in RP, and that's likely because in addition to just GABA A, there's also GABA B and GABA C receptors here, so that just targeting GABA A, which may be important, didn't lead to the same kind of problem as diminished GABA in the animals. However, we reasoned that perhaps kind of super activating these GABA A receptors, like in the brain, may compensate for having a low GABA level in RP. And so we looked at that here, and this is just back to the full field ERG in the RP pigs in red, and here is the wild type in a 250 millisecond flash, and you can see that if we treat these animals with lorazepam, that it really allows the animals to reset this on bipolar baseline and really seemingly largely overcome some of the problems with the reduced GABA. These are quantified down here. Now the half-life of lorazepam is up to 20 hours, at least in patients, and so we asked, what the duration of this effect might be in the pigs, and you can see here this is the untreated pig, the full field ERG, and when we treated with the lorazepam, you diminish this on bipolar signal back near baseline. And then we looked after 4 days and found that this effect persisted And if you retreated with lorazepam, there really wasn't much of an additional effect. However, it's not shown here, but if you go out a week, you basically lose this effect. So this effect can persist for, at least 4 days. So going back to look at the flicker, and this is just basically what I showed you above, that there's sort of an exhaustion with the ability to maintain the flicker with trial numbers. And so the comparison to be made is really here. It's the late trial number in the RP, and you can see that the lorazepam treatment, at least largely reversed some of this effect, allowing the baseline to be reestablished between these rapid flashes, and this is quantified over here. Now, we ask whether or not this signal might be transmitted downstream from the bipolar cells to the ganglion cells. And we think because of the architecture that's involved, where we think that the choroid circulation is critical, the placement of the RP adjacent, and then the adjacent outer retina there, that all that architecture is really important. So we wanted to try to look at what might be going on with the ganglion cells in vivo, as opposed to trying to look at patch clamping or something by puffing, neurotransmitters onto retinal slices in culture. And so when you look at the pattern ERG, which purportedly does allow some assessment of ganglion cells in a checkerboard pattern of reversals here, if you do reversals around 3 hertz or less, you generate this classic pattern of a positive P50 wave and a negative N95 wave. This has been, examined by a lot of people, a lot of different groups here. And if you look at models where you crush the optic nerve, either in primates or in mice, to destroy the ganglion cell signal, you ablate the ganglion cells genetically in mice, or you really increase the pressure by glaucoma models in primates or mice, you really diminish these signals, really suggesting that ganglion cells are important. And Frischman's group have provided evidence, at least in the mouse, that this P50 peak is really largely due to on-pathway signals being transmitted to the ganglion cells, whereas the N95 peak is really off-pathway signals. So this shows what we found when we compared the wild type and the RP pigs. And the black is the wild type pigs, and we see a classic, P50 N95 type peak. But when we look at the RP pig, we see an enhanced P50 signal and really loss of an N95 signal and a positive signal really in its place. One possible interpretation of this is that the constitutive on-cone by pathway signal here is really responsible for this increase in the P50, and for really this positive signal here that's masking the N95 peak. when you look, we then treated with APB to knock out this on cone bipolar signal. And when you look at the wild type, it does significantly diminish the P50 peak. And interestingly, when you look at the RP pigs, you do diminish the P50 peak, and you also diminish this positive signal over here, and you uncover the P95 peak. So we concluded from this that likely this constitutive on-con bipolar signal to the ganglion cells is leading to this sort of a positive PERG signal that's creating this kind of a pattern. Now, the next thing we looked at was sort of contrast sensitivity, and we changed the contrast of the pattern that was presented from 100 down to 2%. And when we looked at the wild type, the PERG really, while you do diminish the P50 signal some by doing this, you still have the same kind of pattern. We then decided that, to look back at an earlier stage of RP. This is progressive, as I showed you in the pig model. So we look back to P60, a time where it's earlier RP. We know that the loss of glucose transport and the effects are progressive. And so when we did this, looked at these animals with the PERG with 100% contrast, interestingly, we saw a PERG that looked basically like the wild type. But as we increased or decreased contrast from 84% down to 70%, we saw that this transition to a positive PERG, and importantly, when you treat these animals with the lorazepam, you basically restore this pattern. So the next thing we did was to look at what we could find possibly about the pathway for GABA synthesis, trying to link that to glucose. So when we looked at incorporation of C13 glucose, we basically found that it was incorporated into the GABA that was diminished in the RP pigs, and we couldn't detect much in the way of GABA synthesis from glutamine, proline, or palmitate, even though those were imported into the retina. So I showed you this earlier, looking at artery versus vein, that we could promote the increase in glucose by treatment with or injection of wild type outer segments. And when we did this, importantly, looking at mass spec, this restored the GABA level in the RP, the retinal GABA level in the RP animals back to a wild type level. Taken all together, this suggested to us that in the retina that GABA is being synthesized primarily from glucose, and by restoring the glucose transport, at least partially, we can restore the defect in retinal GABA back to close to a wild type level. Looking at what might be going on in amacrine cells, and pathways, you can see that there's RNA for GAD expression in these cells, suggesting, and as I showed you earlier, these are really the only cells in the retina, in the human, at least in the pig, that express significant GAD. So we think that they're, they're expressing, they're making GAD from glutamate. And also, if you look at the pathway with the GOT1 and GOT2, suggests that that this may be occurring via oxaloacetate through the TCA cycle. And we have other evidence for this that I'm not going to show you, but this is the pathway, which is not that surprising in the amacrine cells. So to sort of sum this up and kind of put a bow on this, we think that the glucose transport from the RPE to the retina is really important for GABA synthesis, and this is really important for sort of tuning the on bipolar signal and bringing it back down to baseline, which is critical for detection of luminance changes and for visual acuity in the animals. And as glucose is diminished and GABA is diminished in there, this constitutive on bipolar signal, I would propose to you, really turns the cone system almost into a rod-like system, where you have a prolonged on signal, probably with loss of acuity and loss of detection of luminance changes, at the expense. But potentially this could be helpful to the animals as RP progresses, and at least allowing light sensitivity. Okay, so the funding for this is really shown here. It's kind of split between myself and Jen Haidu, also some DoD funding from Yonkin, also RPB and Bright Focus funding. So I'll stop here and entertain some questions, but I just to emphasize to you that this is an ongoing story with this GABA. There's still some holes in it that we're trying to pursue. So thank you very much for your attention.
Thank you, Doug, very much. I have one or two very simple questions. Initially, on the first slides, you showed the glucose accumulation in mice in RP.
Yes.
I assume that this was homozygote knock-in, right?
This is heterozygote animals.
That was heterozygote?
Yes.
And so you have a very quick degeneration in 35 days.
I can say, well, I mean, they're your mice, so I think it varies in our hands depending on the background. As we cross these with black 6, there's some changes. So yeah, I mean, that's what we saw, is that while, let me just go back to that slide since we're talking about it. I apologize. get back here. Yeah, so I mean, by P35, we didn't really see much change in the ONL, but we saw that the rod outer segments were, the length of them were beginning to diminish.
And related to your pig model, those are transgenic pigs, right?
They are, yes. So
yeah,
we
and I
have
should know that. How quickly do they degenerate? What is the
well,
natural history of degeneration?
yeah, so it's fairly rapid. You see that when you look at the outer segments there in the RP animals before birth, they appear normal. After birth, with the onset of light, you can begin to see that the rhodopsin is starting to accumulate in the inner segments, as well as the wild type, rhodopsin from these. There's loss of outer segments pretty much by P20 or P30, and by P60 it varies somewhat. A lot of the rods are gone.
So did you determine how many copies of mutant rhodopsin is expressed in those pigs?
Yeah, there's probably it varies from animal to animal, and the number diminishes somewhat as you passage the animal. But I would say 4 to 5 copies of the mutant. So it's probably, much more rapid and a higher level of expression of the mutant, than you would expect in patients, where you have typically one of the alleles mutated, and it takes, a long time, for the one bad apple, to sort of interact with the wild type and begin to cause problems.
Hi, Dr. Dean. This is Andrew Brown from UC Irvine. Thank you so much for this talk. It really gave us a lot to think about, and especially in the context of pupillographic changes that we see in macular degeneration in our patients, and we'll be thinking about the bipolar cells more. I have 2 questions. The first is related to, the insertion of glucose transporters as triggered by photoreceptor outer segment phagocytosis. Have you, and you mentioned the use of metformin to increase the abundance of those transporters. Have you, in your models, investigated metformin, either intraocular delivery or systemic therapy in the second
No, of course, we're somewhat interested in that. I mean, the metformin that I was referring to is, I think others have are beginning to look at metformin and other, other possible regulators of glucose transport in these models. We haven't, in the pig model, looked at that formally. So the answer is, I don't know. I mean, one might think that based on the diagram that I showed you, that there may be some effect on the transport of GLUT1 to the apical surface and the transport of glucose. But we don't know that ourselves.
Thank you. And then the pattern ERG data that you showed with the use of benzodiazepine, lorazepam, what is the effect on the pattern ERG in a wild type animal?
Yeah.
Have you considered?
There is an effect in the wild type animals. I didn't show that, but it's fairly modest compared to the to what we're seeing in the RP model. Yeah. So there is some effect, some reproducible effect, more with yeah. So there's some modest effect. Yeah. And the effect is to increase the, to really increase the B wave.
Hi, Dr. Dean. This is Philip Kaiser. Very nice talk. I was curious, you showed the slide of the pentose phosphate pathway induction in RPE correlated with oxidative markers in retinitis pigmentosa. Is there anything known about the impact of glucose 6-phosphate dehydrogenase deficiency in modifying the progression of retinitis pigmentosa?
I don't know the answer to that. Somebody else might. So I'm not sure. Someone speak up if you know.
Hi. I have a question about the ERG reduction with subsequent flashes.
Yes.
So you show and you demonstrated that can be attributed to reduced con synaptic transmission. But what about the con photoresponse inactivation phase? If it's slower, that the sign of this effect would be the same. Have you looked at things like expression of RGS9 or similar proteins that are responsible for the con pigment, the con phototransduction and activation, and then before and after treatment with your drug?
Well, I mean, so you can see here, sort of in panel G what we measured. We measured the height, of the B wave here, and we also tried to measure. It's a little tricky with the flicker. We tried to measure the duration of the signal, trying to relate that back to what we saw in the full field. And so, you can see what we saw. There's a dramatic decrease in the height. We think that's because, there's a failure, in the baseline reset, but that's a little tricky to say with the flicker. So anyway, this is what we see with that. And importantly, we think this is progressive. We think this is because that, with enough, subsequent quick flashes that you begin to lose the ability of the recovery, to be able to bring it back down to baseline. At least that's one interpretation out of this. So I hope I answered your question.
Hi, Dr. Dean. This is Vladimir Kefalov here at UC Irvine. I would second the question that was just asked. I think it will be important to rule out any con contribution to this detuning that you see. And my question is actually, again, it's a really striking result that you demonstrate. And I was wondering whether this has been seen in patients with retinitis pigmentosa. Because again, it should be pretty straightforward to do the same experiment with humans and see whether there's detuning there as well.
Yeah. I mean, as you're aware, these patients, while they present with, problems with nighttime vision and peripheral vision, their central vision is frequently okay for a long period of time. However, these patients do classically present with, photosensitivity and with, a really delayed reestablishment of acuity, following a luminance change, and also some changes in color, different color vision. So we think, possibly or we imagine that the photosensitivity and the delay in reestablishing acuity in these patients as, after a luminance change is because of reduced, GABA in there. Yeah, the effect that we're seeing, I have to say, in the pig model on the cones, is typically quicker and probably more extensive than you're seeing, in a lot of the patients. And it's, keep in mind that there's an anatomical difference, in the placement of the cones, in these, certainly in the rodents and even in the pigs, that, versus primates and humans, where they're really concentrated, centrally. Even in the pig, although they're concentrated, to some degree, there's not a macula. And so there may be some, differences in, cone metabolism because they're all grouped together in a macula. So, I dare not say much more than that.
Thank you. Fascinating.
Hi, Dr. Dean. This is Dominic Lewandowski. I want to ask you about what happens with lipids in your retinitis pigmentosa model in the RP. As you showed that there's accumulation of many metabolites in the RP model. Do you by any chance checked if what happens with polyunsaturated fatty acids like DHA or the very long PUFAs? And
yeah, that's a good point. We do have some of that data. We've done, with GenHi, we've used some new plates to do the mass spec coming from Biocrates, where there's now 600 to 800 different metabolites that are available. Some of these are various, length of fatty acid chains and things like that. We're trying to delve into all of that now, but I can't really say much about that just yet.
okay, I understand. But would you expect that there will be accumulation in the RP because they are not able to release transport lipids?
Well, I mean, what I would say is I expect the fatty acids to decrease, in the RPE, in the RP model, because they're deficient in phagocytosing, the fatty acid rich outer segments.
Okay, thank you.
Okay, thank you for this wonderful seminar, and please join me in appreciating the wonderful hour that we had today.
All right. Thank you very much.